Now, how this is going to happen, when the cells are infected with the virus, how these cells are going to make interferons. So here is this image that you can see here. This is a virus that is replicating inside the cell in the red, okay? And this virus is replicating in making RNA, okay? For instance, 5' triphosphate RNA, okay? On its 5', there are 3 phosphates, as you can see here, 1, 2, and 3, okay? Now, this particular molecule, the 5' triphosphate RNA, can be recognized by some intracellular peptides or receptors that are present there inside the cell for the recognition of this particular ligand. Okay? So in case of this ligand, the pink one, 5' triphosphate RNA, there are receptors that are called RIG1. Okay? What is RIG1? It is retinoic acid inducible gene 1. Okay? This is the name of this particular receptor. So this receptor can recognize the pink part or the viral RNA, okay? And for the recognition of this viral RNA, it has got a specialized domain which is the helicase domain. That domain of the receptor RIG1, which recognizes viral RNA, is the helicase domain, okay? It has got 2 domains, this particular receptor. One is the helicase domain, and the other one on this side is the CORD domain, the caspase recruiting domain that we studied in the last lecture. And I had told you that the caspase recruiting domain, normally or in most of the cases, is used by different peptides to recruit caspases and activate them. But these can also be used for joining different peptides together inside the cell. So here you see that this is a CORD domain and this is a helicase domain. Now here is another peptide, a membrane, peptide or receptor present on the mitochondrial membrane or as part of the mitochondrial membrane. This is MAVS, okay? What is MAVS? It is given here. This is the mitochondrial antiviral signaling protein. So it means this is an important part of the antiviral response of the cell, that there is this MAVS present here on the surface of mitochondria. Okay? So what happens that when the helicase domain of RIG1 receptor binds with the triphosphate, the CORD domain of this particular RIG1 receptor recognizes the CORD domain of this particular MAVS, okay, and comes here and binds to this particular CORD domain. CORD and CORD can bind with each other. And when there is binding of the CORDs here, there is dimerization of the RIG1 receptor. And you know that dimer is the active form of receptors. So dimerization here activates this particular signaling cascade in which you can see here are 2 RIG1 receptors and 3 MAVS bonded to the RIG1 receptors with the help of CORDs. And both the RIG1 receptors are bonded to the 5' triphosphate RNA right at this spot of where the triphosphates are with the helicase domain of the RIG1 receptors. And now this is the perfect activation signal to start this signaling for the making of different interferons. So this is the start now. Now this is a perfect assembly that can send out signals. So this activation is going to activate other kinases that you have studied. Okay, for instance, you have studied IKK, or you have studied TBK, for instance, different kind of kinases that would be activated when this assembly or this dimerization is made on the surface of mitochondria, finally leading to the activation of TRAF6. Okay, so TRAF6, this particular protein or peptide, when this peptide is activated, it is going to lead to another cascade of events that finally would lead to the phosphorylation of interferon response factor 3 and interferon response factor 7. Okay, so it would not only be phosphorylated, the pink things attached to their surfaces, which of course some kinases would do, but this also is going to dimerization of these response factors with each other. These are actually transcription factors. And when they are not dimerized, they are not active. When they are not phosphorylated, they are not active. So when they are dimerized and phosphorylated, this is the active form of these interferon response factors. And these factors now are able to be translocated into the nucleus. They, upon activation, will be translocated to the nucleus. Okay, so when they enter the nucleus, they have got specific genes to go there and bind to their promoters. What are those specific genes? For instance, interferon response factor 3, it can specifically bind to the promoter of interferon beta and start the transcription of this particular gene. Similarly, interferon response factor 7, it's going to bind to the promoter of interferon alpha and start the transcription of interferon alpha. You understand this? So these are, this happens when, there is some kind of viral infection, and this happens, the production of interferon by the cell happens in this way. And these interferons are going to do what? They are going to save this particular cell from this particular virus that has entered the cell. Okay? Now these interferons are going to make different kind of products or stimulate other genes or directly interfere with this virus, the viral RNA, or to, there are different means that this interferon production can lead to the destruction of viral RNA. And if it is not possible to deal with the infection, for instance, interferon also are going to help with the natural killer cells to identify this particular cell and kill this particular cell. Okay? So they are using different tactics for that. Okay, so this one is the next one. Now, these are different modes that we have been talking about, that, you suppose that the virus infects an epithelial cell. This is an epithelial cell, okay? And here is the virus that you see here. This virus is coming down and its receptor, specific receptor for its entry is present on the epithelial cell in red that you see here. And when the virus actually, binds to its receptor, it would start a cascade of these different kind of events, So what happens here, that how these virus infected cells are going to produce these interferons, and how these interferons are acting as autocrine or paracrine in different modes. Okay, this is about that one. So what happens on the left panel, you can see that epithelial cell being infected with the virus, and the infection induces intracellular signals that cause interferons response factor 3 to be phosphorylated. Okay, And this induces dimerization and nuclear entry of the interferon response factor 3. Interferon response factor 3 is in green here, okay? So these events will lead to dimerization of IRF3 as you saw in the previous slide. And this IRF3 is going to enter into the nucleus of this particular cell and start the transcription of interferon beta gene. Okay, so this is interferon beta response, but this is going to take the help of NF-kappa beta and AP1. These are other transcription factors that we already have talked about in our previous lectures. Okay, now what happens when interferon beta is produced? This interferon beta is going to take an exit from the cell. It would be secreted, and this is going to act back on the same cell, because there are type 1 interferon receptors on these cells, the epithelial cells. And in the previous lecture, I have told you that interferon beta, it can, bind to, type 1 interferon receptors. So for interferon beta, there are receptors on the cells where they can bind back and, influence the activation of this particular cell or keep it activated. Now what happens in the next panel, if you see, when this interferon beta binds to this particular receptor, you can see that binding here. It is going to activation of another response factor, which is interferon response factor 7. And this interferon response factor 7 inside this cell is going to start the transcription of interferon alpha genes, okay, or interferon beta genes. This one, the green one, is through this mechanism, interferon response factor 3, and the yellow factors that you see here, these are IRF7, or interferon response factor 7, that are going to act upon this particular gene. So interferon alpha will be transcribed, this gene will be transcribed, and the product made would be secreted out of the cell. That is interferon alpha, okay? Now, this is an autocrine mechanism. Interferon beta was made inside this cell, and this interferon beta is going to act upon the same cell with the help of a receptor which is present on the same cells. We are talking about the same cells. Because now, you can take another cells in different color here. These are the yellow panels. These are just describing a single cell, and this one is another panel. Now, what is the interferon beta going to do in case of other cells? The interferon beta binds type 1 interferon receptors on adjacent uninfected cells, giving a paracrine interferon beta response, okay? So if there are other cells in the surrounding, uninfected cells, which needs activation or interferon response, then this interferon beta not only can act upon its cells, the same cell, but also on a different cells which is in the surrounding of this cells in order to activate it and start the production of these interferon beta. Okay, so the mechanism is the same. Via interferon response factor 3, this particular objective can be achieved by interferon. So this is just in words, the same thing that I have shown you in the previous slide, that what happens first, that interferon binds its receptors on the cell. The next thing is that signaling takes place. Once this binding takes place on the surface, the virus binds to the receptors as you see here, okay? So this receptor is either JAK1, for instance, in case of some viruses, or tyrosine kinase 2. These are different receptors. Most of the receptors are of these natures. So these JAK1 and TYK2 or tyrosine kinase 2 receptors, they would send intracellular signals, because they have an intracellular domain. And these signals are going to induce transcription factors, as you saw in the case of IRF3 or IRF7. There are lots of other different transcription factors that can be induced with the help of this signaling that takes place as a result of binding of the virus with its receptor. Okay? Interferon response factor 3 is already there inside the cell, so it is a preformed molecule. It just needs activation. But other, transcription factors should be induced with the help of this kind of signaling. Okay, so one is just activated, and other transcription factors would be induced. It means that this signaling is going to lead to, the transcription of genes that would make different kind of peptides that would act as transcription factors. So what would happen when transcription factors are induced and they are made by the cell? The transcription of many genes, including all type of interferons, and except this one, interferon beta. Okay, except interferon beta, all many different types of interferons that I showed you here in the red, these are synthesized with the help of this kind of signaling or via induced transcription factors. Now what happens that some induced proteins, some of the proteins or the genes that are induced with the help of these transcription factors, they interfere directly with replication of viral genomes. Okay, for instance, the example is here, oligoadenylate synthetase. It is an enzyme and it polymerizes ATP from 2 to 5 prime with the help of 2 to 5 prime linkages. Normally in human nucleic acids, you would not see 2 to 5 prime linkages. The linkages are 3 to 5 prime linkages, but this enzyme just alters that tradition and would just polymerize ATP in 2 to 5 prime linkages in that way. Okay, so just making these viral nucleic acids a target for endonucleases. When there is a 2 to 5 prime linkage, it's not a 3 to 5 prime linkage. This is something alien to the human cell, because there are no 2 to 5 prime linkages in the human cell or human DNA. So anything that is wrong with inside the cell is dealt with by different means, including the use of different endonucleases in order to cut these molecules that are wrongly synthesized, or that are faulty in nature. So what will happen? The different restriction endonuclease enzymes which are present there would be activated in that case, and they would just, cut through these viral nucleic acids inside the cell so that the virus doesn't propagate. So these are the genes induced by interferons or other induced factors with the help of which, with the help of these peptides, the viral nucleic acids are just gotten rid of. Now, interferon alpha and beta also induce protein kinase R. This is another enzyme that is doing phosphorylation. And this protein kinase R prevents production of the viral proteins. In the initial, description, I told you that they can use different ways in order to stop the virus from being replicating inside the cell successfully. And that can be done by just, stopping this process of replication or stopping the process of transcription of the genes of this particular virus that has entered the cell. or by stopping, peptide production, translation, or assembly of the particles. All these are different steps in making of the virus, transcription, replication of its genome, transcription of its genome, translation of the genes that have been transcribed, and assembly of these particles are all different steps that that should be, interfere with the help by these different kinds of interferons or different peptides inside the cell. And in this way, the viral infections would be stopped inside the cell. Now, interferon response factor 7, it can start the transcription of interferon alpha without NF-beta and AP1, and this accelerates production of the interferon during infection. More interferons are needed during infections, and it is achieved with the help of this particular, thing. As you saw here, interferon 7. So interferon IRF7, I'm talking about that. In case of these infections, IRF7 can start the transcription of both types of interferons without the help of other transcription factors like NF-beta and AP1, which are required normally when the virus binds to the receptor, and this signaling actually starts there. Now, what other steps are involved in this process of activation? Interferon also binds its receptors on the natural killer cells. As I told you, all nucleated cells have interferon receptors. All nucleated cells are prone to viral infections. So all of them produce interferons. All of them should have interferon receptors on their surfaces. Otherwise, interferons cannot influence those cells. So natural killer cells also have receptors for interferons. And when interferons bind to these receptors, the natural killer cells are activated. And once these are activated, these natural killer cells are going to find the viral infected cells and kill them. And cytokines draw these natural killer cells to tissues where they kill virus-infected cells. Okay? They are invited to the site of infection with the help of different cytokines that we would study in our future lectures. Okay? So the basic rule is activation of these natural killer cells by these interferons that are produced in this process of infection, and these activated natural killer cells then would go and kill the viral infected cells. Okay? So in the next lecture, I will tell you about other roles of the natural killer cells and macrophages, so I will try to summarize the role of these different cells and, also inflammation. And then we can just move to other parts like we can discuss adaptive immune system, etc. Okay? Thank you very much.